So the next arm you can say is the DHU arm, also called as the dihydrouridine containing arm. So one of the base present there would be the dihydrouridine. That's why it is called as dihydrouridine arm. This would be called as the D loop, and this will be called as the D stem. Similarly, this will be called as anticodon stem, and this will be called as anticodon loop. Together it is called as anticodon arm. So together this is called as the DHU arm. So every arm will have a loop and a stem. Just remember that. Loop will have unpaired bases and stem will have the paired bases. So the DHU arm is the arm which is supposed to carry 8 to 12 unpaired bases, and remember this loop is the largest of all the loops here, and it is the largest loop and has dihydrouridine there. It is the binding site for the enzyme. Which enzyme? Amino acyl synthetase enzyme. That means the enzyme which helps in amino acylation or attachment of the amino acid here. So the next one is the fourth one is the XII arm. So this arm carries the stem and also carries the loop. So together it is called as the arm. So it has 7 unpaired bases, out of which one would be the pseudouridine. Another would be pseudouridine is written like this. I just told you a few minutes back also. Then RT, or sometimes even sometimes even simply T is mentioned there. That is for the ribothymidine. So ribothymidine and pseudouridine are present in this arm. That's why it is called as T-Psi, and then C is present there. T-Psi-C arm. And remember, this arm is the binding site for the ribosome. So ribosomes attaches here. So ribosome attaches here, enzyme attaches here, amino acid attaches here, mRNA attaches here. Now the variable arm. I just told you a few minutes back, it's the extra arm. It is a variable side arm or the loop which lies between the anticodon and the T-Psi arm, between these two. And it has, so far we have understood that it is not present in all the tRNAs, and the exact role of this extra arm is still not known to the biologist. I hope this whole structure is clear to you. This is how tRNA looks like in the 2D. Now compare this with this one. This is how tRNA appears to be in the 3D. In 3D, this is the way it undergoes folding. So 5' end undergoes bending like this, and finally it goes up to this much. So 5' to 3'. This is the place where the amino acid attaches. This is the arm which is also called as the T-Psi arm, to which the ribosome attaches. This is the portion where the enzyme attaches, and this is the portion where the mRNA attaches with the help of the anticodon. So when you open this whole thing into 2D, it appears to be like the clover leaf shape. I hope this whole diagram is made clear to you. Now what exactly are the functions of the tRNA? So the first function of the tRNA is to act like a adapter. I already told you. This was explained by Crick. Crick was the scientist who was the first one to say that the tRNA is the adapter molecule, and its function is to transfer the amino acid to the ribosomes for synthesis of a polypeptide or a long chain of amino acid, or you simply call it as protein. So there are different types of tRNA for different amino acid, and remember, one amino acid is generally carried by one tRNA, but there are exceptional amino acids also for which 2 to 6 tRNAs are possible. This is all because of wobble position. We will be understanding later, not now. Now remember, it attaches. So function of the tRNA is to attach with the messenger RNA with the help of the anticodon arm. Its function is to attach with the ribosome with the help of T-Psi Its function is to attach with the amino acyl synthetase enzyme with the help of DHU arm, and its function is to attach with the amino acid at their 3' end. So this is how the tRNA functions. Now this there is something this brings us to the next topic now, and which can be considered as the messenger RNA. So this is the last type of the RNA which we have to understand. Just remember one thing, that the anticodons are present on the tRNA and the codons are present on the messenger RNA. So how do you distinguish these two things? As anticodons occur on tRNA, codons occur on mRNA, or you can even say codons occur on DNA, because DNA's information is changed into messenger RNA. Then remember, anticodons are complementary to a codon, and codon is complementary to the triplets of the DNA. So just remember this, that anticodons are always complementary to the codons, and codons of the messenger RNA are complementary to the template strand of the DNA. What is template strand? In the DNA structure it will be clear to you. Template strand. So anticodons are complementary to codon of messenger RNA, and messenger RNA's codon is complementary to the template strand reading of the DNA. Then the third point you can remember is that anticodon helps in bringing a particular amino acid at its proper position during the process of protein formation. But when it comes to codon, codon determines the position of amino acids. So anticodons help in bringing amino acid, and codons help to determine the position of the amino acid in a chain. I hope these differences are clear to you. Now this brings us to the messenger